Good afternoon, I'm Ellen J. Lagnas, and today I will be discussing about biotechnological applications and its impact. So what is biotechnology? Biotechnology is often abbreviated to biotech. It is the area of biology that uses living organisms to develop or make products. It is also characterized by long development lead times. It can take as much as a decade to get a new drug from the test tube to pharmacy shelf. For centuries, the principles of biotechnology were restricted to agriculture, such as harvesting better crops and improving yields by using the best seeds and breeding livestock. The field of biotechnology began to develop rapidly from the 19th century. Biotechnology is widely used in different fields such as medicine, agriculture, industry, environment, and others to produce useful products for human benefits. In medicine, biotechnology has a variety of applications, and some of these applications in medicine include the following. First, recombinant insulin. It was discovered that the insulin produced by the pancreas of the pigs can be used by humans. But there were not enough pigs to provide the quantities of insulin required. So this led to the cloning of the human insulin gene. The specific gene sequence that codes for human insulin were introduced in E. coli bacteria. The gene sequence altered the genetic composition of the E. coli cells. Within 24 hours, several E. coli bacteria containing the recombinant human insulin gene were produced, and the recombinant human insulin was isolated from E. coli cells. Next is gene therapy. Gene therapy holds the most promising answer to the problem of genetic diseases. Gene therapy is used to treat genetic disorders, usually by insertion of a normal gene or correct gene for the defective or inactive gene into an individual with the help of vectors such as retrovirus, adenovirus, and herpes simplex virus. The normal gene replaces the defective or inactive gene and carries out its functions, and accordingly the therapy has the highest chances of developing a permanent cure if introduced in the earliest stages of life. Another one is molecular diagnosis. Medical diagnosis is another application of biotechnology in the health sector. Many times the pathogen concentration increases by the time the disease is diagnosed. Hence, early diagnosis and knowledge of pathophysiology are essential for an effective cure. This can be achieved with the help of techniques such as recombinant DNA technology, polymerase chain reaction or PCR, and enzyme link immunosorbent assay or ELISA, and others. The pharmacogenomics has led to the production of drugs that are best suited to an individual's genetic makeup. It can be applied in diseases such as cancer, depression, HIV, asthma, and others. And last, vaccines. Vaccines are obtained by animals and cell structures. These vaccines contain inactivated pathogens. The transgenic plants can produce antigens that can be used as edible vaccines, and antigenic proteins from several pathogens can be expressed in plants such as tomato and banana. Transgenic sugar beet can treat foot and mouth diseases of animals, and transgenic banana and tomato can cure diseases such as cholera and hepatitis B. Next is the application of biotechnology in agriculture. It involves scientific techniques such as genetically modified organisms or GMOs, and examples are Bt cotton, and pest-resistant plants. Genetically modified organisms, or GMOs, are plants, animals, bacteria, or fungi whose genes have been modified by genetic manipulation. So they are more tolerant to stresses such as drought, cold, heat, or others. They are also pest resistant and therefore less dependent on chemical pesticides. Genetically modified crops help reduce post-harvest losses and help to increase the mineral usage by plants, thereby preventing early exhaustion of soil fertility. Genetically modified crops have enhanced nutritional value. Example is vitamin A enriched rice. Now let's look at some examples of genetically modified crops and how they are useful. First is Bt cotton. This is a genetically modified version of cotton, and Bt stands for the Microbacillus thuringiensis, which produces an insecticidal protein or toxin that kills other insects such as tobacco, budworm, flies, mosquitoes, beetles, and others. Scientists isolated the Bt toxin genes from Bacillus thuringiensis and incorporated it into various crop plants such as cotton. This variety is Bt cotton. Since most Bt toxins are insect group specific, the choice of genes to be incorporated depends on the crop and the targeted pest. And the other one is the pest-resistant plants. RNA interference, or RNAi, is a method of cellular defense in all eukaryotes. It involves the silencing of a specific mRNA by a complementary double-stranded RNA that binds and inhibits the translation of this mRNA. Scientists took advantage of this process and introduced nematode-specific genes into host plants using agrobacterium vectors. The introduced DNA produces both sense and antisense strands in the host cells. These complementary strands then produce dsRNA and initiate RNAi and thus silence the specific RNA of the nematode. Consequently, the parasite cannot survive in the host that expresses this RNA, leading to resistance against that parasite. So moving on to industrial application, first here is food and drink. Biotechnologists have designed and built bioreactors or fermenters, which are chambers in which microorganisms are cultured in a liquid or solid medium. Fermentation takes place in the bioreactors and the desired products are either the cells, example yeast, or some enzymes, drugs, alcohol, and others. Next is biofuels. The recent increase in the price of petroleum products have pushed many nations to embark on a desperate search for alternative sources of fuel. The search has focused on using living organisms and biological processes as sources of fuel, especially ethanol and biogas. Next is medicine. The traditional medical products of biotechnology were antibiotics. Advances in biotechnology, especially in genetic engineering, have led to the production of new drugs. And one typical example is human insulin, the hormone responsible for regulation of blood sugar. Next, microbiological mining. Bacterial leaching is used throughout the world as an additional technique of extracting metals from ores, mainly copper and uranium. The bacteria convert insoluble metal to soluble metal compounds from which the metal can easily be extracted. And last, the biological detergent. Enzymes, which are biological catalysts found in living cells, are being used as washing powders. They have the ability of removing stains of biological origin. Now let's proceed to the application of biotechnology in environment. So the following points highlight the four main applications of biotechnology in environment. First, waste treatment. Second, biodegradation. Third, microorganisms in pollution control. And fourth, biomass energy production. So let's discuss first the waste treatment. The nitrogenous waste materials are treated both aerobically and anaerobically alternatively. The degradation of nitrogenous compounds gives ammonia, which is then converted to nitrate by nitrifying bacteria. Then, by the action of denitrifying bacteria in anaerobic environment, releases nitrogen gas in the atmosphere. Second, biodegradation. It is the process by which materials such as oil spill, herbicides, pesticides, and others are degraded by the action of microbial system. Organic compounds, which are naturally occurring or biogenic, are biodegradable, while man-made or xenobiotic compounds may be biodegradable, persistent, or recalcitrant. And third, microorganisms in pollution control. Microbial strains can be isolated in order to control various forms of chemical pollution, such as biocides, detergents, plastic materials, and hydrocarbons. Genetic recombination techniques should make it possible to achieve such a result that a single microbial strain will be capable of decomposing and assimilating numerous compounds, most often non-biodegradable or xenobiotic, produced by the chemical industry. And last, the biomass energy production. Biomass is the living matter or its residues, a renewable source of energy. Biogas formation and methanogenesis are, although ancient forms, today they are in great demand. Methane fermentation is widely practiced and is one of the easily available sources of energy. Agricultural wastes and farmyard wastes are efficiently used for this purpose. Now let's talk about the biotechnological impact. So like all technologies, biotechnology offers the potential of enormous benefit but also potential risks. If we focus on benefits, biotechnology could help address many global problems such as climate change, an aging society, food security, energy security, and infectious diseases, to name just a few. But if we look on the other side, just like other emerging technologies, we cannot predict with absolute certainty the risks with biotechnology. For example, synthetic biology is already contributing very much to the development of many biological systems, producing drugs, chemicals, and fuels without using fossil resources. However, if misused, synthetic biology can generate biological and chemical materials that are harmful to human beings as well as the environment. And genome editing, especially when it is performed on people, will always carry ethical questions. So that's all. Thank you for watching.